All right, today we're going to be doing some sketch notes. So the materials you're going to need to do this will be a piece of paper. If you have blank printer paper like I have right here, that's best. You'll need a pen or a pencil to draw with. You will eventually need some colored pencils. And if you like to be precise, you might want to get something to use to trace a circle. I'm going to use this soup bowl. It's pretty big, but it's perfect for what I'm going to do today. For the next set of sketch notes that we do for mitosis, you're going to want something a little bit smaller like a drinking glass. But for today, something like the soup bowl is perfect. If you don't have something handy, you can just freeform it, and that's totally acceptable. If you need to go grab some materials, pause the video right now, go grab them, and then resume. All right, so the first thing I'm going to do is I'm going to trace, I've titled my paper "This Cell Cycle," and then I'm going to trace my bowl over here. I'm going to put it off here to the left side of the paper, and I'm just going to trace my circle with my pen. Doesn't have to be perfect, but we're essentially making a pie graph. So a pie graph is not used very often in biology, but it's perfect for what we're talking about today, to show a concept, to teach a concept about how a cell spends its life. So cells go through periods where they're just doing their job, but they're also growing and they're getting ready to divide into what we call daughter cells. So anytime a cell divides, the resulting cells are called daughter cells. There are a few exceptions. Some cells never do divide, or they have a very long delay before they go through cell division, but for the most part, cells go through what we call cell division. And we're going to go into all the details of cell division, called mitosis, in our next set of sketch notes. All right, so first thing I'm going to do is I'm going to just go ahead and work on the details of my drawing of my little pie chart. You might also want a straight edge. I'm just going to use this little notepad I found here, and it's going to work just fine. So I put a dot kind of in the center of my circle, and I'm going to draw a line to the dot, and then I'm going to draw another line out here to the side. These are approximate, but I'd say this is what, like about 3 o'clock on a clock, about like so. And then I'm going to go back and I'm going to make a little sliver of this, about 1/3 of that wedge, pie wedge, into a dashed line. So just dash, 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 like that. Okay, so far so good. And then I'm going to divide this larger pie wedge into 3 approximately equal parts, also with little dashes. So dash, 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 and another one about right here. Dash, 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 dash. Okay, there we go. That looks good. Okay, so these wedges are going to represent how a cell spends its life, what it does with its time. And all of this on the outside is really part of one big cycle called interphase. So I'm going to draw a little arrow like so to represent that all 3 of these wedges kind of belong to the same phase, and we'll add some color at the end which will also help visually with that. And I'm just going to write interphase, and I'm going to kind of turn my paper for writing interphase. Inter means in between, so this is going to be the in-between phase in a cell's life cycle. Make sure that looks good. Okay, there we go. All right. Okay, this wedge up here from this spot to this spot represents the period of time in which a cell is actually dividing, making daughter cells. So I'm going to just write cell division and then draw a little line to show that these are both parts of the cell division process. All right, remember you can always pause the video at any time if you need a little more time to get caught up. All right, then M phase is how I'm going to label this wedge. And the M stands for mitosis. Okay, this little smaller wedge, which is also part of cell division, I'm going to label the C phase, and the C stands for cytokinesis. And we've actually studied both of those roots. Cyto, remember, means cell. Kinesis means moving, the cell moving. So it's when the cell is moving apart and separating into two separate parts, into two daughter cells. All right, so this is the actual cell division portion. And tomorrow, or next time we meet and we do a sketch note, we're going to go through the details and the steps, and we're going to draw what the cells look like during this mitosis phase, going all the way into cytokinesis. So we'll go into all those detailed drawings of cells. But we also today just want to kind of talk about what's happening when the cell's not dividing. And so we label these interphase sections into G1. G1 stands for gap 1. And then this middle one is called S. S stands for synthesis. We've had that term before in biology. If you remember, synthesis means to make something. So we're going to be making, the cell's going to be making something during S. And then this last big wedge of interphase is called G2 or gap 2. All right, so let's just add a few little cheat notes on the diagram. So like at a glance, we can kind of remember what each of these three parts of interphase mean, what's happening. So, and it also helps me kind of think of why it's called that. So G1 is growing. So not only is the cell just doing its job, if it's a skin cell, it's protecting you, it's also growing, because as a new baby daughter cell, it's usually about half the size of a fully grown cell. So it's putting some time and energy into growing. All right, and then S phase, we said, stands for synthesis. So the thing that the cell is synthesizing is it's making copies of the DNA, copying the DNA. So I'm going to write that little note right there. Okay, while it's doing that, it's still doing its job, not just copying DNA. So think about, why would it be copying DNA? Well, it's about to divide in half and make itself into two daughter cells. So it's going to need 2 copies of all of the cell instructions. So that's why it's doing that. And then a keyword for G2 is resting. So there's still some work in preparation of cell division happening, but kind of a key term you can think of is the cell's just doing its job, resting up, and getting ready for this really intense portion of its life cycle called mitosis, where it divides into 2 daughter cells. Okay, very good. So if you have some colored pencils handy today, I would grab those. And our coloring today is pretty straightforward, pretty simple. So if you don't have them today, you can always come back and add coloring to this later. But I just think visually, for me, it helps if I can color all of interphase, even though we broke it down into these separate phases, subphases. It helps me, when I look at my notes, to remember that they all do belong together as part of interphase when I color them the same way. And if you pick a light color like yellow, it's faster and easier, and you don't have to spend so much time coloring it. And also you can still read your writing. Okay, then my other part of the cell, I'm going to pick another light color. I'm going to pick orange, and I'm just going to kind of shade cell division, which is mitosis and cytokinesis, M and C, orange, to once again kind of show that they also belong together. It doesn't have to be perfect. Okay, very good. All right, now I'm ready to get into a little bit more note taking. Just off to the right side, I'm going to write a few more notes, and we're going to actually start with interphase. So, interphase. Okay. All right, in interphase, we said the first stage is called G1. So we're just going to write a few more notes, a few more details than what we have up here, so that we remember what happens in each of these steps. So that stands for gap 1. Okay, so remember I said when you hear G1, think growth. So cell growth. Okay. The cell has just divided. So there are a few cells in our bodies that don't go through this portion of their life cycle, or they may go through them at a much later stage or only if necessary. And so there are a few cells that kind of end the cell cycle right here. They actually kind of exit the cell cycle at this point. So let's make a note about that. Some cells exit the cell cycle at this point. Can you think of cells in your body or parts of your body where if you have some kind of damage, it's common that there might not be repair, like people who don't recover from certain types of injuries? So think about somebody who's been in a diving accident and maybe they have damage to their spinal cord, or somebody who damages a muscle, like a muscle gets cut or something happens to a muscle. And so nerve cells and muscle cells are examples of this. So that's why people with spinal cord injuries often either it takes a really long time for their recovery or sometimes they never recover at all. So there's some very cool, innovative new techniques to try to get around that. And using stem cells is one of the therapies that they've used for that, which is still considered to be a little bit controversial. But if you want to find out more about that, that'd be an excellent E.O. Wilson project. Okay, so these muscle cells and these nerve cells will keep doing their job. They're just not going to continue on and proceed with getting ready for division. Okay, the next step is S, which is synthesis. Okay, and so we said that means to make something, so the cells copy the DNA and prepare to divide. So they're still doing their job again, like skin cells would be protecting your body from foreign invaders. But in addition to that, they're copying all their DNA because, again, if they're going to split into two cells, they're going to need 2 copies of everything inside of them so that the 2 daughter cells can be identical. And that is the goal of cell division, is to end up with 2 identical daughter cells. Okay, G2. And remember we said G2 stands for gap 2. Okay, so the cell is resting and doing its job, whatever its job is, in preparation for cell division. So once again, the skin cell is just protecting your body, just hanging out, doing its job, knowing that it has a lot of work ahead during cell division. All right, cell division. Let me make sure you can see everything. I'm going to scoot this up just a little bit so you can see better. Okay, cell division. Okay, so we had M for mitosis. And we said that that is just the part where the nucleus is dividing. So mitosis is all about how do we get half of the information that was in this one cell into one of the daughter cells and half of it into the other equally. So we need to make sure that all the material inside the nucleus, which is the DNA, it's been copied already, but it has to be split up and divided between the two cells. That's the most important thing. And then C stood for cytokinesis, cell movement, and it is where the cytoplasm divides and the cells physically split apart from each other. So remember, when we talk about cytoplasm, we're talking about not just the jelly goo stuff, which is technically called cytosol. We're talking about all of the organelles floating around inside of it, like the mitochondria and the ribosomes and the Golgi bodies and all that good stuff that makes the cell work. And so in cytokinesis, all of those are going to be split up. They had to be copied as well. So it'll be like one mitochondria for you, one mitochondria for me. And so that's where that physical split, division of the organelles, and then the cell physically divides. So the cell physically splits during cytokinesis. All right, so I'm just going to color code this once again. So up here I made this orange, so let's make these orange, kind of show that they go together. And then I'll go up here, I'll do the same thing with interface. And, you know, make it as pretty as you like it. These are your notes. You will turn in a picture of your notes for some points, for some daily points, but then they're going to be your notes to help you on the next unit test. So do what you need to do to make them work for you. If you're a very visual person and you want to spend more time going back and making them fancy, that's totally up to you. All right, so I'll sign off for today, and next time we'll do some sketch notes over just this part, cell division.